Perfect.
So this is in aid of our lab that we're doing on Monday.
Yes. Okay. Can you guys hear me okay? Is the volume okay?
Yeah.
Yes.
Awesome. Okay, perfect. Thank you. Okay. Yes, so I'm going to be showing you some pipetting techniques. So I will start now. So an important part of this DNA lab that we're doing next week is using these micropipettor devices. I'm going to do a close-up of them in a second, but just to give you an overview. So these devices are very commonly used in science labs, and they're great because you can deliver very accurate volumes and a range of volumes and very tiny volumes as well, as small as 1 microliter, which is barely visible. It's so tiny. Good pipetting technique is really important for this lab. Normally we get you guys to practice in advance, but of course we're in a different time now. So I'm just going to go through the basics of how to use these. And then I'm hoping when you guys come in Monday, I'll still have a little bit of practice stuff set up so you can practice while there's a few steps where you're waiting. So you'll have a chance to practice yourself as well. Okay. So I have here, there's a range of them. So there's 4 different ones. The lowest range goes from 0.2 up to 2 microliters. So again, that's very tiny, barely visible. Then we have a 2 to 20 microliter, a 10 to 100 microliter, and one that's 100 to 1,000. Okay. So let me just switch cameras here and I'll show you a closer view of these guys. There, okay, awesome. Okay, so let's start with this little one here. Okay, so just to show you the basic parts. So these have a dial here that tells you the volume that you're going to deliver. And you can adjust the volume using this wheel. Okay, so you always have to understand what the range is of the pipetter before using this, of course, because this 10 could mean all sorts of things. But in this case, because we know the range is 2 to 20, then we know that's 10 microliters. Okay, so the first thing you would do, so let's just say I want to pipette, let's go for 15 microliters. So I would just turn the wheel here till I get to 15, and you can do actually volumes in between too. So you could do like 13.5 or whatever custom volume you want. We'll just go for 15. There we go. Okay, and then the next thing you need to know is what kind of tips to use. So there's different types of tips for different sizes of pipetters. This particular one, I believe, would use the small size tips, these little ones. I'll show you the other one too with the larger tips. Okay, and the way these are arranged, these are often used in labs where you want to, you're dealing with very sensitive samples. So you want to keep the tips clean, and you also want to avoid yourself touching the samples. So they're arranged in a way that you can grab them without touching them. So that's why they're in these packs. And usually this whole thing is autoclaved to kill off any sort of resident bacteria that's living on the tips. And that way we know they're really clean. Okay. So I'm just going to go like that. And now I have my tip. Okay. Then that. Okay, so I have some solutions here that I'm going to use just as a practice. I'll use this one here. It's just some red glycerin. And I'm going to suck up the solution using the pipetter. So to just suck it up, you have to press down on this part here and then pull it back up. Okay, and this part here has 2 settings. So when you push down, you'll feel it stop. That's the first setting. And then if you push really hard, there's another setting. So when you're drawing the solution up, you just go to the first setting and then pull up. If you're dispensing the solution, that's when you go to the last setting to just get the last tiny little drop out. Okay, so you don't want to go all the way when you're pulling the solution up, because then you'll pull too much into the pipetter. Okay. Okay, so I am depressing the plunger thing and then sucking the solution up. Okay, so you could see it there in the tip. 15 microliters is still a pretty tiny amount, but it's enough that we can see it. Okay, and then I'm going to put it into this tube. And this is something you'll have to do this a number of times during the lab. You'll be putting little tiny drops of stuff into tubes, and most of the stuff you're using is clear. So here we have the advantage, we can see it because it's red. In your case, it will be clear. So often what we recommend you do is just put the drop on the side of the little microtube, unless otherwise instructed. That way you know it's actually in there. If you put it all the way down to the bottom, you might not know it went in, and also you might contaminate the tip. So it just depends. Again, sometimes it'll tell you to put it right in the bottom, but if not, then just put it on the side like that. Okay, so I can see it there. And again, when I released it, I pressed all the way down to that last setting. If I didn't do that, you would end up seeing a little bit. Actually, let me try that. Okay, so press down. So I haven't pressed all the way, and you can probably just see, might be a bit hard. There's a little bit left in the bottom. So to be accurate, you need to get all of that out. And then the other thing here, before you let the plunger off or release it, you need to get your pipette out of there, because otherwise you're just going to suck up the solution again and just undo what you just did, which would be sad. So don't do that. Just pull it out and then release it. Okay. All right. There's my nice
I think
solution.
that has happened in the past.
There's all sorts of things happen during this lab, but I'm sure you guys will be perfect. So then we have our tip, and we're going to assume this is contaminated. In your case, you will be dealing with your own tissue. It's a very, very tiny amount of your own tissue, but it's still, we want to treat it like a biohazard. So we don't want to touch the tip, and also if there's chemicals on it or whatever. So there's a special release lever here that will allow you to just take the tip off without touching it. So you just press that, and then the tip should, oh, sorry, it's not working for this one. Oh yeah, because this tip's too small. So in this case, I might have to pull it off, but I'll have gloves on. Okay, most of the time it will just come off when you do that. And usually we will put it in the biohazard container. Okay. Okay, so I'm just going to try that one more time, but with a very tiny volume, so you can see how hard it is to see. So I'll go all the way down to like the bottom setting. Actually, no, I know what I'll do. I'm going to get this little one here. Okay, so this one's already down at 1 microliter. This one goes from 0.2 to 2.
That is so tiny.
It is, yes. And there is a step. I think right at the very end, you do all this stuff to your DNA and get it all ready. And then you have, like, it all rests on the success of getting 1 microliter of a solution of an enzyme in with the DNA. So yeah, but I'm sure you guys will successfully do it. Okay, so there we go. Just gonna suck that up. Ooh, yeah, okay. So it's a very small amount, you can barely see it. And now I'm going to put it into the tube. Let's see. And here it's really important to put it on the side. So I can see there's a little blob there, so I know it went in. Yeah, so it's all good. There it is. Okay, and now I can get rid of this tip. And, in the lab, when you do this, so you're often adding a few different things in here. So again, you'll want to avoid cross-contaminating with the pipettes. You'll put it on different sides. Then usually there'll be a step where you'll try to get that all down to the bottom, and you can use either a centrifuge or a vortexer or tap it. So in this case, I'm just going to tap, and there we go. It kind of went down, most of it. That's why a centrifuge is actually a bit better. Okay, so now everything's down there ready to go. Okay. Now, now I'm going to show you how to pipette into the gel, which is kind of another really important part. There. So this is actually, this is an old gel from last year, but I can just use it to practice filling. Okay, so we can actually see, well, there's actually student DNA on it. So can you guys see? Yeah, I think you can see there's, actually, I'm just going to pin my own video here so I
Yeah.
can see what you guys are seeing.
You can see the bands, the
Excellent.
DNA
Yeah.
bands. Yeah.
So this is the ladder here. So that's where like the calibration row basically, or column. And then I can actually see a bit of student DNA there. It's a bit faint for you guys, but there is some there. So this step here is the sort of the end. You've got your sample already. And now we want to separate it out so that we can actually do some measurements on it. So what we do here is load your sample into these wells here. So there's these little wells that have been created here. So this is like agarose, so it's kind of like a jello type substance. I melt it and then pour it into a mold, and then it solidifies to sort of a jelly type consistency. And then while I'm doing that, I put a little comb in here to make these little pockets. Okay, and now the other thing is it's underwater too. So you have to have it in a buffer when you're pipetting into it, which makes it extra tricky, but that's okay. So I'm going to be putting, I'll put 20 microliters into this. Just adjusting my pipetter. And a tip about the pipetters, sometimes people accidentally adjust it over the maximum volume, and you don't want to do that because then it just gets stuck and stops working. So just make sure you're always double checking what the range is before you do anything with it. Okay, so I'm going to grab one of these larger yellow pipette tips. And now the good thing with these solutions is we often add a loading dye into it. And the loading dye has a couple purposes. One of them is so that you can track the progress of your sample in the gel. The other purpose is to ensure that the little sample is a bit heavier than the water so that it will sink to the bottom of that pocket. If you didn't have that, you could the sample could just kind of go floating away, and then of course you wouldn't see any results. Okay, I'm going to suck that up. And now I'm going to put it into one of these pockets. Oop, that one's a bit closed. Here we go. There.
You are such an expert.
Glad it worked. I was under pressure there.
Well done.
I shouldn't test my luck and try another one. So, but I'm going to do one, but I'm going to show you what can go wrong. Okay, so if you don't get it into the pocket, you kind of go like, dun, 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 it's gone, right? That's it. Again, you've done all that work, and then your sample's just gone. So that's why I'll make a few practice plates. So again, you should practice this step before you actually have to load anything, just so that you know what you're doing. And the other thing that can go wrong is if you go too deep down in these wells, you can poke through the bottom. So say I do it there, and then the sample's gone. It just left through the bottom of the well, of the gel. So you have to get it just right. You just put it about halfway down through the well and then release it. Okay. All right. So I think that's good. Is there any questions about all this or anything else you want me to show you?
That was fantastic.
Yes, well, I know it's not the same as practicing yourself, but as I said, you'll have a chance to do a bit of practice next day when you come in. And
Cool.
yeah.
Thank you.
Awesome. Okay.
Could we, for Monday, could we pre-make the gels? Well, I'm asking you actually, are you going to be pre-making the gels? Because I know that's an extra step. Might take
no,
too long.
I can't, but I just realized the problem is that I'm probably gonna have to do the gels for you guys because